Okay, man gave names to all the animals. This little PowerPoint is just in existence to point out that this question of where we place all of this life on the planet and what little cubby holes in what classification niches has to be based on certain properties. And usually, at least the early systems of classification were based primarily on what the organism looked like. So we're just going to go through a few of those means by which we classify. Some of them have become far more sophisticated in recent years, where we look at primarily molecular similarities in the DNA between species, but a lot of these other tools are being used as well for classification. So let's start at the beginning. The song itself, of course, is kind of interesting because it points out perhaps a more artistic sense in which man gave names to all the animals. When we talk about names, of course, we're talking about scientific names, that genus and species names. You might find, here's a little question for you, the song that's playing in the background. See if you can identify who is actually singing. All right, moving on. Normally we look at organisms and classify them based on their structural similarities. All animals with gills and scales we basically like to call fish, right? Well, not necessarily. Looks alone can actually be deceiving here. A porpoise, this guy right here, looks very similar to the blue shark, but guess what? The fact is the porpoise is in fact far more similar to the Holstein cow. Both are mammals. The blue shark is in fact a cartilaginous fish. This structural similarities, you may even extend them to things like wings. Birds and insects have wings, but insects, these guys, are not at all related to birds. The point here is that it is important when classifying organisms to go beyond simply appearances. Birds and flying insects do in fact look similar, as do the porpoises and the blue sharks, but no, classification depends more on a series of taxonomic tests. Things like analogous structures. Analogous structures are adaptations that possess similar features and functionality as a result of exposure to a common selective pressure, but have different underlying anatomies due to having unrelated evolutionary origins. So an example is the formation of the streamlined body shape in aquatic animals, shark, penguin, and dolphin. These body forms are streamlined regardless of ancestral origin. Example, the sharks are fish, dolphins are mammals, penguins are birds. What this illustrates is something called convergent evolution, as unrelated species have become structurally more alike due to exposure to the same shared selection pressures. They all tend to live in similar environments. So analogous structures are often confused with what is called homologous structures. But in homologous structures, these structures possess a similar underlying anatomy. In this case, it's called the pentadactyl limb, penta, 5, dactyl, in vertebrates. So these are said to all possess a similar or shared evolutionary origin, but have evolved into a variety of distinct forms due to the presence of different selective pressures. They have, if you will, different functions. So as I mentioned, the typical example of homologous structures is this pentadactyl limb structure in vertebrates, whereby many animals show a common bone composition despite this limb being used for different forms of locomotion. So example, the whale fin is used for swimming, bat wing is used for flying, human hand, you might say, manipulates a whole bunch of different kinds of tools, and the horse's hoof would be used, and of course there's no horse's hoof here, but could be used for galloping. What this illustrates is something called adaptive radiation via divergent evolution. And we'll get to the whole evolutionary stuff later in the course. But again, for the purposes of classification, analogous structures, homologous structures, these are various taxonomic tests that we use to put organisms in their particular taxa. So the more similar the homologous structures between two species are, the more closely related they are likely to be. So what about reproductive patterns? Dogs, cats, insects, and fish have a lot more in common with each other than they do with, say, daffodils, mushrooms, bacteria, and paramecium. So they all belong in this same taxonomic group we call kingdom Animalia, because they are all heterotrophic. None of these are capable of photosynthesis like plants. They can sit out there in the sun all they want, and they're not going to be able to make food. They have to go shopping, they have to go hunting, they have to eat from a sort of different troughs, so to speak, heterotrophic. Notice, however, that these organisms are not capable of reproducing each other. Your cat is happy about that. Right. Even as you move through from phylum to class to order and beyond, you'll notice that your family dog has much in common with members of each group. For instance, a spinal column composed of bony vertebrae in the case of phylum Chordata, the potential to nurse young, which makes her a mammal. Of course, we're talking about the dog here. In fact, the dog is a meat-eating mammal, which of course makes her a carnivore. Note, however, at this general level, reproduction is still impossible between the dog and the other organisms in the order. Point is, species and subspecies are the only levels at which all domestic dogs can interbreed and produce fertile offspring. That is basically the definition of a species. The word specific points to this specialization at the species level. This is another taxonomic collection of various ways we group life on the planet. We basically look at physiological, nutritional, and cytological evidence. You've done a little work on biology vocabulary, so you know that cyto means cell. Cytological evidence, we're primarily looking at, is the kingdom made up of organisms with cells that are prokaryotic, or are they eukaryotic? So where a living organism fits, first into a kingdom and then into the various taxa within each kingdom, very much depends on the kind of information in this table. As you move through the course and you study each of these 5 kingdoms, Monera, Protista, Fungi, Plantae, and Animalia, the things that you will hopefully learn is just this information in the table. You, for example, are made up of cells. You're an animal, no offense, made up of cells with discrete nuclei. You are therefore eukaryotic, and therefore you belong to a domain called eukaryota. Since your cells lack chloroplasts and cell walls, you are a multicellular heterotroph, there you go, with highly differentiated tissues and organ systems. This makes you a member of kingdom Animalia. Let's just say this chart is worth coming back to when you're looking at basically mode of nutrition for each of the different kingdoms, cell wall type, cell type, etc. Good stuff. There's another sometimes used taxonomic test called embryological development. Not so much used anymore, but it's given a whole lot of credence historically. This is simply studying the growing embryo in animals and plants, and what it tends to show is that closely related organisms go through similar stages of development. So you recall, after fertilization, you develop into this embryo, and the embryo develops further, and as you go through each of these stages, notice that there are a whole lot of commonalities. All terrestrial animals, for instance, have non-functional gill slits, which are called pharyngeal slits, as early embryos, which to a lot of people suggests that they have some sort of aquatic origin. Many vertebrates, including humans, demonstrate a primitive tail at certain stages of embryonic development. And the last one. This is probably the most important one in terms of where we are today. You are, of course, looking at the double helix of DNA from different organisms. Molecular evidence involves identifying conservation in DNA and protein sequence as a basis for determining evolutionary relationships. So you'll recall that DNA is the code found in the nucleus of your cells. It is the recipe for all of the proteins in your body. You're made up of proteins. Basically, these recipes spell out who you are. So one technique used to compare DNA between organisms is called DNA hybridization, and of course there are other DNA techniques as well. But anyhow, as you know, DNA is double-stranded, but if you heat it, it's actually going to separate and will eventually reform or reanneal as the temperature falls. Single-stranded DNA from different species can be mixed together to identify the degree of similarity. And again, this is as measured by complementary base pairs. Remember way back in, I think it was Science 9, guanine pairs with cytosine, cytosine pairs with guanine, adenine pairs with thymine. So those base pairs will align correctly when these single strands are placed together. Closely related sequences will join together, that's called hybridize, more strongly as they share more complementary base pairs. The strength of the hybrid molecule and the degree of similarity can be measured by how much heat is required to separate the strands. Closely related conserved sequences will have a higher melting temperature, Tm, than distantly related sequences. So the fact that these two strands basically pull apart at low melting temperature suggests that they are not as strongly held together, not strongly related, whereas these two organisms seem to be more closely related because even at higher temperatures, their DNA strands tend to stay together. And that is how man gave names to all the animals.